Assalamualaikum and a very good day. In this video lectures, I would like to continue our first chapter by discuss about this slide. Okay, you may refer to this slide, which is a digital image processing, digital imaging fundamentals. In this video lectures, we will cover these 5 items. Okay, however, the one that I would like to emphasize more, it's about the one that's important in this topic or in this lecture is this one: image representation, image sensing and acquisition, and the one which is the one that is we'll discuss more is about the sampling, quantization and resolution. Okay, however, the first two topic you can read by yourself. Okay, I just explain a little bit. Okay, of course, if you talk about the image processing, something which is the technology itself, it's mimic the human behavior and human characteristic. Of course, when the cameras nowadays, when the camera was founded, actually they also try to mimic the behavior of human, especially in human visual system. So which is why if you see the camera, actually the inspiration how they develop the camera is come from the human visual system. Okay, so this one you can read by yourself. Okay, this is I think this one have been covered in your previous course. Okay, which is in biomedical imaging, you also discuss about this one, which is your bandwidth or your electromagnetic spectrums. Okay, consists of many spectrum or type of electromagnetic spectrum. However, the one that can be seen by naked eyes is a visible spectrum or visible light spectrum, while the rest cannot see by human eyes. Okay, such as X-ray, gamma ray, and so on. So the one that you will do image processing is related to visible spectrum or visible electromagnetic spectrum. Okay, which is why like I discussed before, such as your X-ray machine. Okay, your X-ray machine, the end part of your X-ray machine is called intensifier. Of course, inside the intensifier, still the intensifier itself will receive the X-ray spectrum. Okay, the signal which is in form of X-ray electromagnetic spectrum, which is X-ray, which is cannot see by eyes. So that's why in that part, intensifier will consist of some characteristic where it will convert that X-ray electromagnetic spectrum into visible light electromagnetic spectrum, and that visible spectrum actually is analog image, and the end part of intensifier, which is CCD camera, or I'm sorry, a CCD sensor, that is the one that will convert from the visible spectrum, which is in form of analog image, into digital image. Okay, so this is some basic understanding about how actually image generated, how the visible light, how something in your environment can be seen by your naked eyes. Okay, for example, if you look at this green object, so why you see that object is green that is because that cylinder actually, okay, inside this slide, this cylinder have a characteristic of reflecting the green spectrum while absorbing the rest of the visible light spectrum, which is why you can see the object is green. So similar to other objects, such as object, let's say a red ball. Okay, that red ball has a characteristic of absorbing the rest of visible light spectrum while reflecting only the green light spectrum. So which is why you can see by your eyes that or the red ball have a capable to reflect the red spectrum while absorbing the other visible light spectrum, which is why if you see that red ball by your naked eyes, you see that ball is red. Okay, this one have been discussed before. Okay, again, like I said, anything in your environment can be considered as analog image because that is the one you seen by your naked eyes. And then of course you need to have an imaging system. For one or a part inside the imaging system should have a capability to convert the analog signal into digital signal. That's why that is the part where actually the imaging system will convert your analog image into digital image, which is why you can get or obtain your digital image. Okay, the imaging system here you can assume like your digital camera or your medical image modality such as CT scan, X-ray, MRI, ultrasound, and so on. Of course, again, why if you can see in this figure, it's not just depend on the object itself. You also need to have the illumination source. In this case, you can say that your light is your illumination source, or you can say a sun is your illumination source, because without illumination source actually you cannot see that object, you cannot see the object, which is why again if you close your light, you see everything's dark. Even a red ball, you cannot see that ball is red. So that's mean illumination source is very important in order for you to see what actually the color or the spectrum which have been reflected by that object. Okay, which spectrum have been absorbed by the object, which spectrum have been reflected by the object. The one have been reflected is actually the color of that image, the color of that object itself. Okay, which is why you see that object red, that object blue, that object green, because the object have a capability to reflect that spectrum to your naked eyes while absorb the others visible light spectrum. Okay, again, this is the one that is important to be discussed, which is sampling, quantization, and resolution, which is the last topic in this slide. Okay, we will talk in detail about what is sampling, what is quantization, and resolution. This is the important things to be focused on. Okay, in this class, start from this topic to the last topic of this course. We will more focus on one channel image. Okay, one channel image or single channel image. Okay, if you remember what we have discussed before, type of digital image, it can be categorized into three, and in some node it categorized into two. One is a single channel image and another one is a multiple channel image. The one which is a multiple channel image is the example of multiple channel image is like your color image, because the color image consists of R, G, and B, which is a three channel image. While the single channel image, the example of a single channel image is like your grayscale image as well as binary image, because both of them actually come from a single channel image. So which is why start from this topic until the end of this course, we will talk more about the single channel image, which is either grayscale or binary. Okay, so if you see, this is how image have been represented. Okay, that's why start from the previous topic, we always discuss about the matrix, because or in form of pixel. Okay, if you remember. Okay, if you remember in the previous slide note, which is this one, okay, we talk about single channel image and multi-channel image. So of course a single channel image consists of two type. One is a black and white, another one is grayscale. So as you can see, why we call it a single channel image, because it consists only one matrix layer, while the multi-channel image, okay, it consists of three or more than one matrix layer. So that's why they call it a multi-channel image. But from here you can see that we already discussed the image itself, in terms of something like a metric form. Okay, because you can see each and every box here, we call it as a pixel, and each pixel contains the color. Okay, what I mean by color, is it black, is it white, is it a very light gray, is it very dark gray, and so on. And this color itself, okay, the color inside this box, or what we call so-called as a pixel, actually, if we convert it into numbers format or figure format, it can be seen as a set of matrix. So that regardless is it grayscale or black and white, you can see this like a matrix forms there, which is why digital image is actually can be represented as a matrix. Okay, back to the our current slide, which is this one. So one way to represent a digital image is you can represent as a matrix. It can be have a column and row and so on. Of course, inside the row and column you can have a numbers, which is like your matrix. Okay, it's like a matrix. It is matrix, and where the position of each matrix itself, each element actually represent the pixel, while the numbers actually represent the color or the intensity of the image. Okay, again, we talk about the single image or single channel image. So if you talk about the grayscale image, you can go the numbers inside this matrix you can have from 0 to 255. Okay, what is the reason behind that? Okay, we have discussed previously, because of each element will you can have or the resolution or the intensity resolution there is 8 bits. That's why you can have numbers from 0 to 255. Okay, this one will be discussed later in the next slide. What is the difference between why we have this range of number, why is 8 bit, why is a 4 bit, and so on. Okay, that will make you clear later in our next discussion. Okay, this is the same things, the process of image acquisition, where you need some analog image, some illumination source, and then the CCD sensor inside your imaging system will convert that analog signal into digital image. Of course, it will map into small box there. The small box there represent a pixel. That's why it have been digitized. Okay, now after, inside here, what happened inside the imaging system, and before you produce the digital image. Okay, so there's two things happen. One is called sampling, another one is called quantization. So I believe that you have been exposed to these two terms in your previous course. Okay, because what I understand that before you having this course, you also have to learn you have to learn biomedical signal processing and signaling system. So I think this one also have been exposed inside your biomedical signal processing class. Okay, because the biomedical signal processing class, it's handling the one-dimensional digital signal. So of course that also at that things also talk about what is quantization as well as what is sampling. Okay, never mind, let's recap back what have been learned before we go deep discuss about this sampling and quantization. Okay, I managed to draw something here. Okay, just to recap back what have been learned um, before I show this whiteboard that I draw. Okay, let's go to this picture. Okay, this one. Okay, this is just some terminology to try to I just try to relate between biomedical image processing and the previous class that you have attended. Okay, I think during the earlier of your study you have been taught about signal and system, where actually you learn about the analog signal, and then after that you take, I think last semester you take biomedical signal processing. Okay, and might be if you are from a school of electrical engineering, you will learn digital signal processing. Actually both are similar, they talk about digital signal. Okay, so from analog signal I just draw some sinusoidal signals there. From analog signal, you then when you go to the biomedical signal processing, you learn about digital signals. However, both are same in terms of the dimension, both are one-dimensional signal. And in this class, okay, you will learn a two-dimensional signal. Okay, two-dimensional digital signal. That's why you can see that I draw in form of two-dimensional. You have x, you have y, and then the impulse, or I'm sorry, the amplitude of the stamp function there, or the impulse response there, is your f. Okay, same goes to this one. This one, the amplitude is your y, but this one, the amplitude now is f, because f now should have the two coordinates or two horizontal axis, which is x and y. So that's why they call it a two-dimensional digital signal, where you will learn in this class. Okay, biomedical signal processing. Of course, in previous school they also learned digital. They also learn this course, but they call it the course they name it as a digital image processing, while in our program we call it as a biomedical. Sorry, this one should be biomedical image processing. Okay, image processing, not biomedical signal processing. Okay, so I hope this overview can help you in relate what you have learned later in this course with your previous course. Okay, so now let's discuss about the quantization and sampling. Okay, in one-dimensional signal, okay, this is how the sampling and quantization looks like. Okay, I believe this one should be covered in your biomedical signal processing. So again, this is your analog signal that you learn in signal and system, while when you have a sampling process, actually you are converting, okay, the continuous number or the continuous numbers with respect to the horizontal axis. When I said horizontal axis, is this one. Okay, it's this one. Yeah, when I said horizontal axis, is this one. This is your horizontal axis. Okay, so sampling will convert, or will convert the continuous number or the continuous signal with respect to horizontal axis, which is why you can see after sampling you have a discrete value. Okay, you have discrete value of the horizontal axis part. Okay, that is sampling. So now how about the quantization? Quantization is the process where after you apply the sampling, this signal will go into this part. Okay, this is the same signal. You don't have to worry about it. This is the same signal. Now what is quantization? Quantization is a process of converting the continuous value of your vertical part. Okay, when I say vertical part, which is this one, okay, into discrete value. Okay, so that's why you see over here, that's why in you know, in converting the analog signal into digital signal, you need both process, sampling and quantization, because sampling is only, you know, try to convert this continuous value which respect to the horizontal axis into discrete value. So that's why you see there's a noun is a discrete there. There's only you only have this number and this number, but you cannot have something between that number. So that's why now you have a discrete part of your horizontal axis. But how about the vertical part, okay, or y part there, y axis there? It's still a very continuous numbers there. Okay, it's a continuous number, which is this number come from here. So that's why after having digitization process of the horizontal axis, now we need to proceed with the quantization, because quantization will digitize the vertical part. So that's why now, okay, let's say this is the point that you can have value, means that between this red line, the red dot line should not have any numbers, because now we want to digitize that point. Okay, that's why we only have this point, this point, and this point. So that's why after you do some quantization, okay, every point which is not which is not in line with that dot line, it will, you know, set as the one that nearest to that dot line. So that's why you can see this green, for example this one. Now because this one is close to this one, so at the end it become here, it going to that point. So that is the what I can explain about what is the thing happen, what is the meaning by quantization and sampling. Meaning sampling is converting the continuous value to discrete value with respect to horizontal axis, while quantization is converting the continuous value to discrete value with respect to vertical axis.